Today's lesson is going to be all about the process of replication of a molecule of DNA. We'll talk about protein synthesis on another occasion. DNA replication is something that we mentioned briefly at the end of the previous topic, where we were looking at the cell cycle. You'll remember that just prior to the process of mitosis in the cell cycle is a long period called interphase. And one aspect of interphase is where the chromosomes are replicated. So in our case, we have 23 pairs of chromosomes in our body cells, and that replicates itself to 46 pairs just prior to the process of mitosis. So at the end of mitosis, when that's all split in half, our body cells have got the right number, once again 23 pairs. So we need to spend some time looking at how molecules of DNA are replicated. But prior to that, we need to look basically at what the molecule of DNA looks like. You can think about a molecule of DNA as a ladder, with molecules joined end to end down one side and again down the other. And other molecules link the ladder together. And then the molecule of DNA, it's almost as though somebody takes the top of the ladder and twists it and turns it round. And you end up with a double helix shape, where the ladder has been twisted and coiled around itself. So there's the rungs of the ladder. And it forms a double helix, a helix being a coil. And each of these two backbone strands makes the coil. Now we need to focus a little bit. What is the coil actually made of? What is this made of? So we'll look at just a section from there and look at the structures of the molecules concerned. What we're looking at here are things called nucleotides. Which we've come across briefly before. We looked at ATP, adenine, adenosine triphosphate, ATP. So a nucleotide is a relation to ATP. It's basically made of three components. It's made of a sugar, which is a 5-carbon sugar. It's made of a phosphate group, and it's made of a base. And the bases are one of 4 different things: adenine, thymine, cytosine, or guanine. We'll come on to the differences between those in a minute. But basically, a strand of DNA is made of large numbers of these units, a pair of those running all the way down. So these form the cross-links of the ladder, and the sugar and the phosphate form the backbone, the pillars of the ladder. So there are the rungs, there are the straight bits of the ladder. Now, how do they fit together? Basically, the molecule of DNA fits together because of 2 aspects. Firstly, the structure and size of these molecules, these bases, and secondly, how they are linked together by hydrogen bonds, which again is something we've looked at before. So, before we go into the detail of exactly which base on one side of the ladder fits against which base of the other side of the ladder, let's look in simple terms at how the process of DNA replication actually takes place. So that's the first thing that happens. The double helix, which I mentioned just now, unwinds, so it's rather like unwinding the ladder, straightening it up again, unzips, comes apart in the middle here, using an enzyme, and so you end up, to begin with two separate strands of DNA. One there and one there. DNA, of course, as you know, occurs in the nucleus. So in the nucleoplasm, the fluid part of the nucleus, there are a lot of molecules of nucleotides floating around, ready to be turned into new DNA. In order for the enzymes concerned to recognize these nucleotides as being the correct ones, they have to be activated. And they are activated by adding extra phosphate groups to the nucleotides. This is something that comes in other parts of biology as well. Other biological molecules are fairly inactive. Things like glucose is not a particularly reactive molecule, yet it's incredibly important in all sorts of biological reactions. And just like in this example, glucose has to be activated in order to make it react with other things, but that's for a future topic. So, extra nucleotides with extra phosphate groups added to them, and that activates the nucleotide. Here they are. So here are the nucleotides. There are 4 different ones, each one representing either adenine, thymine, guanine, or cytosine. And here are the 2 extra phosphate groups added to that, which is basically telling the enzymes that there are nucleotides ready to work. The bases of the activated nucleotides then pair up with complementary base pairs on each of the old DNA molecules. A complementary base is one that will fit next to another one. You can't just randomly line up adenine, cytosine, thymine, and guanine and expect them to join together. They won't, because they're not all complementary to each other. So there are a few things that you have to learn here. Adenine is complementary to thymine. And cytosine is complementary to guanine. So you can't fit an adenine next to a cytosine because they're not complementary. You can't fit cytosine to thymine because they're not complementary. This is the only way they will pair up. And that has got something to do with their structure and also the way that the hydrogen bonds fit together. Now, before we go into the nitty-gritty of how they join up, it's worth just looking a little bit more at what makes these things complementary. Adenine and guanine belong to a group of chemicals called purines. Cytosine and thymine belong to a group of chemicals called pyrimidines. Structurally, purines and pyrimidines are different. Purines are made of a double ring, basically a double ring of carbons stuck together are purines. Pyrimidines are a single ring of carbons. It's obviously more complicated than this. There's other things attached to them, but basically you need to know the difference. That purines are big molecules, pyrimidines are smaller molecules. Although it seems a slightly cack-handed way of doing it, I tend to remember the names by thinking, well, there's a short name with a big molecule and a long name with a small molecule. That's the way my brain works. So that's one of the differences, the sizes. There's another difference. Structurally, when adenine and thymine link together, they link together by the assistance of two hydrogen bonds. So here is an adenine molecule, I'll use this one, and a thymine molecule next to it, and it joins together by two hydrogen bonds, and that forms the cross-link of the ladder. Okay, so this is the cross-link of the ladder. The other two, when guanine and cytosine join together, and again, guanine is a purine and cytosine is a pyrimidine, this time they have three hydrogen bonds. So that's a reason why only one of the purines can join to only one of the pyrimidines. Not only is it to do with the shapes of the molecules, but it's also to do with the hydrogen bonds. Once again, let's go back to the concept of the ladder. Here's the ladder. These are the sugars and the phosphates of the nucleotides. These things have to go in between. So, let's try and put two pyrimidines together. There's one. There's the other one. You've got a big space there. The molecules are far too far apart to allow hydrogen bonds to form. So that's never going to work. Hydrogen bonds are weak and they only join things that are very, very close together. So that's a big gap. Let's try putting purines together. There's one purine. How are you going to fit another purine in that space? The molecules are simply too big to go into the spaces. So that's not going to work either. There's no way is that going to work. So always you have to have a purine and a pyrimidine. And they join together with either two or three hydrogen bonds there or there. So you can only join adenine with thymine, and you can only join cytosine with guanine. So the structure of DNA is largely controlled by bonding and the sizes of the bases. And it's rather like a jigsaw puzzle. You can't stick the wrong shaped piece of a jigsaw puzzle in, it won't physically fit. And that's exactly the same with a molecule of DNA. So, going back to our molecule, we've got the separated strand of DNA. So it's unwound into two strands. An enzyme, DNA polymerase, links the sugar and the innermost phosphate groups of next-door nucleotides together. So these free-floating nucleotides with their extra phosphate groups line up next to one that it is complementary to, and then the sugars and the phosphates in the in the nucleotide link together using this enzyme. And then, because there was the extra phosphate to activate it in the first place, that extra phosphate drops off and can be reused to make another activated nucleotide later. So there's the process. Here's the open strand having unzipped. Here's the open strand having unzipped. Here's a new molecule of DNA joining on. Here it comes. Activated nucleotide, that one will match up to that. So it fits. This could be adenine and thymine, or it could be cytosine and guanine. It fits together. And because it is complementary, this will come in here, and then the enzyme DNA polymerase will link that to that, forming a joint. And so it's basically stretching the ladder up. So DNA polymerase will only link an incoming nucleotide to the growing new chain if it's complementary to the base on the old strand. You might remember when we were doing the cell cycle, we said that it's a very accurate process. There's actually an enzyme that zips its way up and down the newly formed chromosome to check for mistakes. But mistakes very, very seldom happen because of this business of having to join together correctly, adenine only joining with thymine, cytosine only joining with guanine, so the base sequence is almost inevitably going to be right. And the chances of a mistake are very, very slight. Somebody's come up with the suggestion that it might be 1 in every 10 to the power of 8 links which might go wrong. That's like saying 1 to every 1,000 million base pairs that join together, it might go wrong. So it's a very, very accurate process indeed. So what we've looked at is an idea of having a strand of DNA which then separates, and the separated parts form new strands. So where there was one molecule of DNA there's now 2 molecules which are identical to the original one. How do we know? How do we know that this is the system? How do we know that there's not some other mechanism? This is something that you can go into in your textbooks on pages 117 and 118, where there were some experiments done by a couple of scientists called Messelson and Stahl, who worked out that this was the method that was used by carrying out a series of experiments. In theory, there was a couple of other possibilities. It could be that a new strand, a new double strand, just miraculously grows next to the first one. So this one is identical to that one, but what could make it identical? This is more straightforward. You can see, if we talk about complementary base pairing, there's only one way that these things can fit together, which is the right way. But if you've got lots of activated nucleotides all sort of randomly joined together, how on earth could you be sure that that one is going to be the same as that one? Then there was another idea, even more outlandish, that nucleotides might just, or chromosomes might just break up into molecules of little bits of nucleotides. And they all join together to create a new one that was the same as the original, which is even less likely. However, you do need to know that the possibilities exist. So conservative replication will be where one completely new double helix is made from an old one. That was what I showed you just now. Somehow a double helix is just built alongside an existing one. There's no complementary base pairing, there's no knowing the sequence of the nucleotides. Somehow it was going to happen. This one is the one that Messelson and Stahl proved to be the correct one, where each new strand contains half the old original one. And I'll show you a picture of that in a minute. And this was the last idea. Each new molecule will be made of old bits and new bits scattered randomly through the molecules. Very, very unlikely, and nobody could come up with any sensible suggestion how that might work. So this is what it looks like. Here's the original strand on the far left. There's a strand of DNA. The idea of conservative replication, which has been discarded, was that the original strand is still there, unaltered, and somehow a brand new strand forms next to it, which is going to be identical to this one. And nobody can come up with a suggestion as to how that might work. Here is the semi-conservative replication. Semi, in this case, meaning half. So half of the old strand, the red bit, there you are, and half is new. And that all works because of complementary base pairing and the way that pyrimidines and purines linked together by either 2 or 3 hydrogen bonds, as I discussed just now. There's only one way it can work. So this seems to be the most sensible anyway. And here's this random idea. A little bit of the old one there, a little bit of new stuff. Bit of old, bit of new, bit of old, bit of new. How's that going to ever work? Where's the system? Where's the logic in that? This is a logical sequence of events. And so semi-conservative replication is the way in which it's considered to be. In the next lesson, we'll talk first of all about the idea of the genetic code and how all your chromosomes have a sequence of bases, sequence of nucleotides, which are called genes, and how that all fits together. And how we know that a gene is composed of a series of nucleotides. But that is for another occasion.